with developing diagnostic and therapeutic radiopharmaceuticals. And my role in ITM as the global product manager for radioisotopes and devices is also in product development and project management, really related to the isotope development and their devices, but really within the respect of the Pharma Solutions, so within the sales team. So I'm a member of the sales team, and I also interact quite heavily with our sales team in trying to support them with, say, technical questions or any kind of, say, queries or questions that, of course, you or your customers or our direct users may have related to our products. So, no problem. So I picked this, of course, this very controversial title purposefully, and when we talk about most promising future isotopes, of course, it's very important to define what is your criteria. If you were to define criteria as, okay, what are the most promising future isotopes really in a preclinical setting, or what are perhaps the most future promising isotopes in an unmet, very specific medical need, these will of course look very different. And the criteria that I tried to define this as was really what is perhaps from the ITM perspective, and how we kind of predict or how we foresee perhaps the global demand changing, of course, over the next couple of years. So of course, when I discuss these future isotopes, take it from the perspective that I myself am not a medical doctor. I study radiopharmacy and radiochemistry, and of course from the ITM perspective. So it's been mentioned, of course, many times, yesterday and also a little bit today, that with these kits and this kit development that is currently ongoing related to gallium, it's obvious that there really is an increasing amount, and this is really just a super short explanation for what you can see on gallium-68 on PubMed. So gallium is not new. It's not like a revolutionary new nuclide that has only existed in the last couple of years, but it's really starting to take prominence in the academic environment. And of course, in many of your regions, or in particular, say, customers that you serve, you might have a different experience, where you might see, well, in our current customer base, technetium is the most common radionuclide, or F-18. And there's of course many reasons why this could be based on different, say, technology or price-sensitive regions. But if we just look at the global perspective, we can see a lot of the different, say, big pharma companies, of course, with Novartis, their development of local methods is nothing new to you, what you've heard here. But I think it's a really good reflection of where the industry will move. And of course, you all know that we're selling gallium-68 generators. We have our ITM model that we've made for many years in Munich, and it was also shown yesterday in the excellent presentation from Radiomedics. We're working in cooperation to essentially produce this new gigant model that we hope to also, really put ITM's footprint in many different markets with our own, gallium-68 generator that we couldn't access previously with our Munich generator. And we also discussed yesterday at some depth that we really foresee there is going to be an increasing global demand for lutetium, and this is reflected in the different radiopharmaceuticals that are being investigated, the new diseases that aren't anymore this orphan drug status. So there's really going to be it's a really exciting time for nuclear medicine and of course lutetium, and how ITM is planning to really meet this unmet need. And of course, we want to be able to supply the market not just now, but to secure our future. And gallium and lutetium, they're really interesting because although they're of course very different, they synergize in a really interesting way. That you have gallium that is really specific as a diagnostic, and it pairs with lutetium that has this theranostic properties. It's primarily a therapeutic radionuclide, and but it also has the SPECT emission, so this gamma line that you can do SPECT imaging, and you can do potentially some dosimetry. I know there's a lot of discussion today already between Professor Delbasand and Philip Harris about how maybe the industry is moving towards this dosimetry approach and the different opportunities that can offer. So it's interesting in this regard. But I think when you look at radionuclides or these radioisotopes, you have to also look at what are the proven examples, like where is it actually proven to work. And gallium and lutetium are in this field that they each serve a very specific individual purpose for their own radiopharmaceutical, but like I said, together they synergize really well. And I think when you're kind of considering, or you see many manufacturers of these different radionuclides or radioisotopes, and they're propagating many, let's say, messages around picking the right isotope, or why should you pick isotope A instead of isotope B. And I think it's always important to mention that you really have to look at the individual inherent value of the radiopharmaceutical. So if you're designing a diagnostic, it should serve its diagnostic purpose. If you're designing a therapeutic, it should serve its therapeutic purpose. That if you're addressing an unmet need, then you should serve that need. And in this regard, so why is it that perhaps gallium and lutetium, they are these proven examples? Well, one major reason, of course, is the favorable decay properties of both of them for this purpose, but also it's the availability. You know, a radiopharmaceutical is only as useful as the availability of the isotope itself. You know, you can see sometimes for many years that there's people publishing maybe something in a mouse model, and they say we've shown this excellent work, but if you can't get routine access or availability of the radioisotope, you will never get this clinical translation of the radiopharmaceutical. And with gallium, of course, you all know that you have the option of generator or use cyclotron. So it kind of offers this balance of them both, where you could have, for these infrequent users, you could have a generator on site in perhaps a more rural area and you still have access to this diagnostic agent. Or if you're in perhaps, say, a very built up urban center and you could perhaps have a cyclotron or of course a 100 millicurie generator, you still have large access or large availability to this critical diagnostic radionuclide. And with lutetium, as we've spoke about yesterday in some depth, because you can produce it in this nuclear reactor, you know, and as you all know, you can get lutetium, NCA of course, in extremely high like massive activities. We can get it in GMP quality, and you can get it in a really excellent specific activity. So it's essentially, it's massive quantity available, it's GMP, and it has extremely high purity with regards to the radioisotope purity or also the chemical purity. And with both the cases, they've been, you know, studied for many years, and you really have very well understood chemistry of gallium and lutetium. There's so many chelators out there that, you know, perhaps for many of you, when Dr. Lucas Greifenstein spoke here yesterday, he was speaking about chelators that probably many of you have never even heard about, and there's all the time, new people are developing more and more chelators to try and really design radiopharmaceuticals for gallium and lutetium. But you know, one thing that's also really interesting about them is that you're not dependent on these super specialized production centers. You know, although ITM, of course, is developing this expansive radiation network, and we told you yesterday of how we're essentially leveraging this commercial reactor in Canada. But for example, in some places, like I can give you one good example, would be like Triumph in Canada. They're doing extremely interesting work in terms of research, but because they have this massive cyclotron, and it's a government-run facility, the radionuclides that they produce are not necessarily always available. But from a research perspective, they're doing absolutely amazing work. I cannot claim that they're not. But when you think about what about the radionuclides that they're producing, maybe what is the availability from this source to the customer, obviously to their customers, to the patient, and meeting perhaps this patient treatment need. Then, you know, we spoke also yesterday about alpha, both from the ITM perspective and our plans, and also Dr. Lucas Greifenstein also spoke a lot about alphas. And you know, we said, okay, so ITM is developing our own production methods to really, you know, supply the globe. To really, you know, we see the need, both from, of course, your perspective, the academic perspective, the industry perspective, and we think could be, or will be, of course, a very successful radionuclide in our portfolio. And like I said, we're working with the CNL, so the Canadian Nuclear Laboratories, to really achieve this goal, to really make sure that we can provide as much actinium as we are currently doing and beyond with lutetium. And again, this was just a super short introduction to, you know, the promise that actinium is, you know, has. And Philip Harris spoke a lot this morning about, of course, there's a long way to go, but it's the first step. But at least in publications, of course, in many cases, some of these are very anecdotal success stories, and we still need more clinical trials going away to fully figure out, okay, well, where is the exact niche field, or what is the exact treatment or the patient selection that is really going to benefit from actinium-225. But there really is a lot of interest, you know, and it shows huge promise. But again, you know, if you pay attention sometimes to LinkedIn or to different kind of marketing, you know, posts or press releases, you might get the impression that, okay, is beta dead? Does no one care anymore about lutetium, and is the entire market moving towards actinium? And I would say, well, no, because in many cases, perhaps actinium or alpha therapy's true power, true potential, could potentially be reached in tandem, or kind of how it synergizes or how it works together with lutetium. And Lucas Greifenstein also kind of discussed this a little bit yesterday, how they were doing some tandem therapies in their clinical experience, and it seemed to be highly effective. And there's many cases of where they see a patient who first receives lutetium, they don't get any results, and then they carry on with actinium therapy. And Philip Harris was also showing some images related to this today. And again, it's all promises at the moment, but I think it shows huge potential of where the market is going. And of course, why perhaps do they synergize so well? Again, is that you can really consider them to be complementary therapeutics. I don't think that actinium is gonna replace lutetium, because it serves really a very specific niche need that essentially we need in this field of nuclear medicine therapeutics. And of course, like I said, with lutetium, it's huge. It's available in absolutely massive activities. There's a large variety of precursors. And with actinium, currently the major problem at the moment is just supply. There's lots of manufacturers are really working on trying to increase their availability or the activities that they have of actinium-225. And naturally, as more and more becomes available, it will essentially allow more trials to develop and really see, okay, what are the conditions that we should be using actinium in? Where do we get the highest amount of efficacy? But in any case, there is a lot of preclinical work also going on and developing a lot of new precursors. And currently, it is available in very high specific activity from this thorium cow route. And like I mentioned yesterday, ITM, we're developing this cyclotron route, because we think that we can get very high activities, that we're really satisfied with the purity you can get from that, because we don't have any long-lasting actinium-227, which is really perhaps the biggest concern with many people, perhaps from medical physicians and also the regulatory environment around actinium-225. So it's still this synergistic effect that is, well, I would say that at least anecdotally has been published, or that people are seeing in their clinics. A lot more has to be really done, but it shows huge promise. Then this is also, it's a really interesting topic, I think, in the field of radiopharmacy, and it's choosing the right pair. So you have, particularly say with copper, you have many isotopes available of copper. So people are kind of inherently attributing the success of maybe one of the radiopharmaceuticals and extrapolating this, then, well, because of the success perhaps of copper-64, because it has DetectNet, copper-64 dotatate is an approved product, that of course we can naturally extrapolate this success to copper-67. We can just replace one isotope with the other, and we essentially are turning a diagnostic into a therapeutic and assuming that we get this highly successful product. And that's not necessarily the case, In fact, like I said already, we've seen already the clinical effects, or, and this is shown through the approved products of gallium and lutetium. And of course, we believe actinium has huge promise, both in a synergistic therapeutic effect, but even in ITM's own portfolio, we have this combination pair, like Philip Harris showed it earlier on, of this theranostic pairing of F18 with actinium-225. And we think that, like coming back to what I mentioned at the beginning of this talk, you really have to evaluate each radiopharmaceutical for its own inherent value. You have to think, okay, what is the purpose I'm trying to address with this pharmaceutical? Is it a diagnostic agent, or is it a therapeutic agent? And you cannot really just extrapolate that, okay, well, because we have a very good 64, that I'm just going to transform it into a therapeutic copper-67. And if we take a little bit of look at the inherent properties of these two isotopes, so copper-64 is really interesting, is that it is really undeniably is a theranostic radionuclide, meaning that it does have this beta plus, so you can use it for PET. It has an Auger emission, so you get this very high energy LET, that you really get this, say, cancer cell killing at the very short range. And then you also have this beta minus that essentially offers, let's say, short to medium range cell death. And the half-life, again, you cannot necessarily say, okay, a therapeutic must have a half-life over or under a certain amount. That it really has to match the biological half-life of the vector that you're essentially attaching it to. But there was always, for many years, copper is nothing necessarily new. I've seen some studies that, or some publications that, of course, copper-67 production goes back as far as the 70s. And copper-64 has been around for quite a long time. And there was this decades-old problem about what is the stability in vivo of copper. This was always, I think, one thing that was really holding it back. And also is the availability. So how you actually make it. Again, coming back to this question of if you have a really good pharmaceutical, how much of it can you really make, or how much of it can you really provide in the clinical setting? And with copper-64, of course, you can make it in a cyclotron. So any area that has a cyclotron can make it. And with this, say, reasonably long half-life, you can distribute it. So, in copper-64, of course, it's an approved product. So it does, at least in the radiopharmaceutical world, serve a very specific need, and it's proven. So I'm not trying to dispute that copper-64 doesn't have added value. But I think it's really important to consider, okay, just because copper-64 is approved and has added value, we cannot extrapolate this to copper-67. And copper-67 is quite interesting because it also, we have this question of availability. It's often said, well, one of its benefits is that you can make it from different isotopes of zinc. But actually one of the inherent problems of zinc is that it has a relatively low melting point of, just to put into context, 450 degrees Celsius, whereas nickel has a melting point of 1,500 degrees Celsius. So but how does this translate to availability? It essentially means you cannot really do very long radiation times, or you cannot really produce these massive activities that you need for this kind of global supply, and really say to support the commercial phase of the commercial launch of an approved product. And recently, and actually with this copper-67, from looking at the literature, I found there's at least 8 different production routes. So you have lots of possibilities of doing it, but there's always a caveat. It's either, okay, you get very low specific activity because you can't separate it from other isotopes of copper, or it comes perhaps with, say, very low yield, or it comes perhaps with really varying problems, and you would have to look at each individual of these 8 methods to really dig into it in detail. But recently, perhaps what is considered the most promising method of copper-67 production is through this linear accelerator. But still, from what I've seen, the highest number that I found was that they essentially could produce about 2 curie at end of bombardment. But this doesn't factor into, of course, you have to do your processing, you have to purify it. So after all this is done, they said it takes approximately 1 half-life. So currently they're producing about 1 curie. And of course, it opens up the interesting question, how much do you need for a patient dose? I think this is also not defined for copper-67 radiopharmaceutical, but it doesn't really offer much value, I think, in terms of it's difficult to get, and it's difficult in availability, and I'm not sure if it can really have the same production scale up like we're seeing with lutetium. And of course, there's a lot of interest at the moment around the 2 lead isotopes. Lead-203 is also quite interesting. It has a relatively long half-life, but that's not necessarily a bad thing. Again, it always comes back to what is the purpose? How are you using it for? It offers pretty interesting value, actually, if you want to use it as a, again, I'm going to be very careful what I say around predicting dosimetry, but because of the much longer half-life of lead-203 versus lead-212, which is a 10.6 hour half-life, you can really see, okay, how does your radiopharmaceutical behave over time? But of course, this is considering that you keep the conditions the exact same. You have the same peptide mass, because these can result in different changes or differences, essentially the pharmacokinetics. And I think lead-212 is really interesting. There's a lot of say publications coming out at the moment, and if we discuss for a moment the availability, again, it's a really interesting kind of issue, because thorium-228, it's a byproduct of nuclear energy waste. So actually there's a lot of it available. Probably the biggest challenge, I think, personally, of lead-212 is the availability in the form of a generator. So it is really the only feasible route is either you have a thorium or a radium generator. And thorium has a very long half-life, I think of about 1.7 years. So usually they're not making many generators of this. So the most common one is the radium-224 generator. But this has a short half-life of 3.6 days. So it means your generator itself only has a shelf life of about 7 days. So any patients who would potentially receive a lead-212 radiopharmaceutical, you really have to group them together, and there's of course some challenges associated with this. And also with this, the first decay product of this radon generator is radon gas, and again it's an alpha emitter gas. It's a challenge. It's not something, because of course there are lead generators available, but it offers difficulties in terms of scale. So the largest generator that I have heard so far of a lead-212 generator was about 700 MBQ. And another major concern, in my perspective, is this gamma line in between thallium-208 and lead-208. So there's a 2.6 MeV gamma line in one of the daughter decay products of lead-212. Meaning, from a generator point of view, of course this decay process is always happening. So if you have this large amount of activity in a generator, it's always decaying into this, producing a huge amount of this gamma line. So it has the penetrative power of a gamma line with the stopping power in the range of an alpha particle. So this is quite a concern, but again, you can essentially overcome this with adequate shielding. But you might need to redefine what is currently considered the standard of care for, say, in a radiopharmacy, how do we have a safe radiation protection environment? But it's a different kind of question. But I think also, personally, in a radiopharmaceutical itself, because you have this high gamma line, it essentially means that this will be irradiated outside of the tumor region, generally, because of the high energy of this gamma line. And the question is, where does it really irradiate? Where does it ionize? And of course, the most probable location would be in the high density tissue in a patient, or it could even be outside the patient, actually. So if you have a patient who has a very, say, reasonably high dose of a lead-212 radiopharmaceutical, they themselves are very radioactive. They're actually probably irradiating lots of the environment around them. So it's an interesting question, but again, it always comes down to what is the purpose of this radiopharmaceutical. That if it's serving a very important unmet need, or if it's showing clinical efficacy, then it's perhaps this is one of the caveats of this is what you have to live with. So we come back to this question of what is the perfect pair, or is there a perfect pair? Is there such a concept? And this is really like a general statement that of course there are some caveats to this, but ideally your diagnostic agent should provide high contrast of tumor to healthy tissue, and your therapeutic agent should have high tumor accumulation and low organ at risk accumulation. And Philip Harris also spoke about this morning, that perhaps one of the major concerns from actinium therapeutics is the buildup in the salivary gland and the destruction of the salivary gland. That patients perhaps they can't go through the full regime of the scheduled treatment because of the high dose accumulation in these sensitive organs. And this question of if I have my diagnostic, I can use this perfectly to predict the behavior of my therapeutic. Again, it's not always the case. That essentially, your diagnostic should serve one purpose and your therapeutic another. And if you change the conditions, like for example, the amount of peptide from this peptide mass, it will for sure behave different in the body, and they have different pharmacokinetics. So of course, then you have to be very careful in assuming how your therapeutic will behave based off of this diagnostic. And of course, then the big question of availability. If the success of your therapeutic is based off of your diagnostic, or vice versa, it would be a real shame to have this, really nice therapeutic developed, but of course, if you don't have the adequate diagnostic that is required to essentially facilitate the treatment of this therapeutic, then of course you can't use it in a patient. But of course, there's many different ways that, there's different reasons that perhaps a patient would receive a therapeutic, and it goes beyond just having a diagnostic companion. So yeah, it's really important to remember that, that two isotopes of the same element are not necessarily the ideal diagnostic therapeutic pair, and that that you really have to look at the inherent value that the diagnostic and the therapeutic can offer you. Then, when we're speaking about actinium, and we're speaking about ultra-lead or astatine, there's a lot of this talk about alpha emitters at the moment. and Philip Harris mentioned it this morning a little to a moment about what about you get this free daughter nuclides, and it adds this difficulty in dosimetry. Lucas Greifenstein spoke about it yesterday, about when you have these decay products or these daughter nuclides, it makes the quality control quite difficult. But I think it's really important to put this into context. So first off, why does this happen? So in the case of lead, you have this Auger emission, and this 36% is referring to this study here from 1992, where they did it with DOLA, where they found the Auger emission of lead, it ionized the chelator and essentially released then all the decay products free in solution. But it's important to remember, this does not mean it will be like this for every chelator. You have to really investigate each chelator, what is its stability with lead-212. That's just an important point to mention. But in any case, for example, when you go from lead-212 to thallium-208, you have this alpha emission. So this path here happens 36% of the time, and essentially the emission of this alpha particle creates so much kinetic energy that it kicks the nuclide out of the chelate, irregardless of the chelate, pretty much. And in the case of actinium, it's the first one that happens. So I think it's important to put this kind of into context, because perhaps many of you, if you're traveling to congresses, I've seen this mentioned so many times that, okay, well, if you use actinium-225, you're injecting 3 or 4 times free activity into the patient. And I think this is really misleading, and I think actually it's deceiving, and I think it's probably more harmful to the alpha therapy community at large. So I'd like to kind of take a moment to put it into context. So if we take the example of actinium, so we see this blue line here is essentially, this is a 2-hour time frame. It's the ingrowth of the daughter nuclides over time. So if you take it to, say, the situation of perhaps a commercial radiopharmacy. So we're sending actinium to they do their labeling on site, they're doing their quality control, then you would have, say, shipment to a patient site, and then you have this injection of the radiopharmaceutical into the patient. So you essentially have this build-up, because this decay process is always happening. It's not something that you can selectively decide to start, okay, we're about to inject it into the patient, now let's start the decay process. Of course you can't control this, it's always happening. So what about the daughter nuclides, And it's, again, very important that it's difficult to predict, of course, dosimetry. And I'm not gonna try to do that now, but I'm trying to put it into context of a way that I think I can communicate clearly. Is if we look at what is the remaining activity, perhaps post-injection. So this example here would be, this is in becquerel, so this would be about 7 MBq, which would translate to about 0.19 millicurie. Of course, I'm not trying to suggest this is the optimum dose for an actinium radiopharmaceutical. It's just kind of an educated guess that I'm using for this example. So if we look at what happens perhaps in the first 12 hours, that you can see that 93.3% of the actinium, so the activity, is still bound for targeted therapy. It's still bound to the vector, to the antibody, or to the small molecule that you want to deliver to the tumor. So, of course, depending on the accumulation time of your radiopharmaceutical, that you actually get the majority of the activity delivered to the tumor. That, in fact, after, in this case, at 24 hours, this should be this is a typo, it should be 24 and not 12. So after 24 hours, you've less than 0.1% of the free activity remaining. And it's important that, consider, of course, it's an MBq, but of course it translates perfectly to curie as well. Like 7 MBq of actinium is not the same as 7 MBq of, say, the first order nuclide francium, and it's not the same as 7 MBq astatine. So if you inject this, say, radionuclides into the body, they have completely different therapeutic potential because of the half-lives of the radionuclides. So then what if we look at perhaps the total amount of disintegrations or the total amount of decays over time? So I took 1 half-life of actinium as an example, and I stuck with the same activity. So if you inject, say, this actinium radiopharmaceutical into your patient, that you can see over time the vast amount of disintegration or decays come from the targeted alpha therapy. They come from the actinium that is bound to the chelator once it's injected into the body. So again, over 99% of the, and this is just in a 10-day window. Of course, if you have, there's a lot of talk about agonists and antagonists over that today and yesterday, that if you have a radiopharmaceutical that can deliver your payload, so in this case actinium, to the tumor, you can get this, really overwhelming amount of decays are occurring in the tumor. But of course, it depends on the washout of your radiopharmaceutical. But of course, as you extend the time, this number just gets higher and higher and higher, getting closer to essentially 100%, but of course never absolutely reaching it. And I include this here again, because it's really important to think about it, to remember that 7 MBq of actinium is not the same as 7 MBq of francium or any of its daughter nuclides. So perhaps when people are propagating this message of like, where you're injecting free activity, or 4 or 5 times free activity, it's really misleading. So I think it's important to remember this. Then this is really just to put it all into context, that for example, with an actinium labeled radiopharmaceutical, post-injection, 99% of the total decays are occurring from targeted alpha therapy. So the actinium that is still bound to whatever peptide or molecule that you have it labeled with. And I think coming back to this question of the most promising radioisotopes in the future, like I said, it comes from the perspective of ITM and perhaps me as a radiochemist, but we're because of this, essentially, we know, or we're seeing this demand in lutetium, and we're seeing that it's going to be only going to get higher. There's too many clinical trials going on. There's too many companies developing, all these different lutetium-based radiopharmaceuticals, that the market is just going to essentially increase. The demand of lutetium is increasing globally. This will not slow down. It's only going to really increase in a log fashion. And and we predict the same will be for actinium. But really, like I said, you have to really define where or what disease state or what indication is your actinium going to serve. And this is still, it's under question. Exactly how can we say really optimize the synergistic relationship between lutetium and actinium. Of course, there's many clinical trials going on. The question is still being answered. Again, there's a lot of anecdotal evidence to show it has a lot of promise, and we're very optimistic for the future. And then if we come back to again, what are the radioisotopes of the future? We see with lutetium, there's an ever increasing amount of indications and diseases that it's been used for. There's new radiopharmaceuticals being developed. With actinium, we see there's a lot of research going on in terms of trial. And also at ITM, of course, we're doing our best, and we're really working towards increasing the production capacity that we have and ensuring really steady supply. And and how these molecules all work together. That we think, sometimes the term theranostics is thrown around quite a lot in different contexts. Sometimes people say that a radioisotope must be theranostic because it has therapeutic and diagnostic capabilities. Or perhaps it's more of just a systems approach to medicine. That, for example, you have the theranostic approach of gallium lutetium, say dotatate, or gallium lutetium dotatate. But we think that the synergistic effect is really what is going to drive gallium lutetium and actinium as probably being the most successful commercial radioisotopes in the near future. So with this, I would like to end my talk. If there's any questions, I'd be more than happy to answer.